Today we will do an experiment on isolation of DNA and do a study on what is isolation of DNA, who is the first person to isolate DNA, what are its applications, materials required, procedure and observation. Now, what is isolation of DNA? DNA isolation is a technique used to extract DNA in a biological sample. In this experiment, we isolate the DNA from our saliva. Does our saliva actually contain DNA? The DNA in the saliva originates from the cells that are shed from the inner linings of the mouth and from white blood cells. These DNA containing cells are collected and then DNA is extracted by various methods. Who was the first person to isolate DNA? Friedrich Mischer in 1869 isolated DNA. What are its applications? To study the genetic cause of diseases, for the development of diagnostics and drugs, to find DNA sequencing, to carry out DNA tests essential for carrying out forensic science. The materials required for the experiment are saliva, 70% isopropyl alcohol, food color which is completely optional, dishwashing detergent, salt, glove for taking necessary precaution, stirrer and a spoon. For the first step, spit into the clean container until it is quarter full of your saliva. Now, for the second step, we take the saliva and add 2 to 3 drops of dishwashing detergent into it. The purpose of adding dishwashing detergent is to break open the cells by a process known as lysing. Now, using a spoon, add some amount of salt into it. The purpose of adding salt is to clump the DNA together. Now, using a stirrer, mix the ingredients well. Now the next step. This step is a completely optional step wherein we add 2 to 3 drops of food color into the alcohol and mix well. The purpose of adding food color to alcohol is to distinguish alcohol from water. Now for the last step, we take the ingredients mixed saliva and the food color added alcohol. Tilt the container containing the saliva. Using a spoon, dribble alcohol to the sides of the container. Wait for 2 to 3 minutes and you will see precipitated DNA. This is the DNA that got precipitated when we added the alcohol to the saliva. How does this DNA get precipitated? This is because DNA is insoluble in alcohol. This is the spooled out DNA that we observed.